Okay. Told myself to remember to do that before. Those online, can you hear us now? Yes, we can.
Yes.
Yes, finally can hear you. Thank you.
Sorry about that, everybody. All right, so let's talk about multiomics, and specifically, I'm going to talk about single-cell RNA-seq plus ATAC-seq. ATAC-seq is something really cool that I'm gonna talk about in a minute. First of all, RNA sequencing. So RNA sequencing is a relatively new technology, maybe the past 30 years, but essentially we can read RNA from cells. This is through a combination of like chemistry and microfluidics and magic, and I don't really understand it, but we can read RNA. And if you remember the central dogma of molecular biology, DNA leads to RNA, leads to proteins. Proteins are the molecules that do stuff. And so if you know which RNA molecules you've got, then you have a pretty good sense for which proteins you've got, and that makes for a pretty good readout of what a cell is doing at any given moment in time. So what we can do is we can find that messenger RNA and read it, and then map it back to the genome and say, here are the levels at which these different genes are being expressed. And so when we're working with RNA sequencing data, we're typically working with counts. How many times did we observe a transcript of that gene in a given cell? And this is a super simple example, but usually we've got on the order of tens of thousands of cells, and you've got about 25,000 genes in the genome. So this leads to a really huge data set. Crucially, it's very, very sparse because most genes are not used in most cells. So you've got about 90 to 99% sparsity here. Most of this matrix is zeros. Similarly, we've got ATAC-seq, assay for transposable, assay for transposase accessible chromatin, that basically just looks for any part of the DNA that's open and accessible. Because most of your DNA is super compacted because it needs to fit in a tiny, tiny cell. Not many regions are going to be open and accessible to proteins that would come in and bind the DNA. ATAC-seq measures which regions are accessible, and you end up with reads like this, that are sort of piled up in random places around the genome. Sometimes they correspond to genes and sometimes they don't. And so we don't have the nice features of genes that we had before. Typically what we have to do is go in and run some sort of peak calling algorithm to find the regions where those reads tend to accumulate. And then we'll use those peaks that we identified as our features for analyzing this data. And so then it behaves very much like RNA-seq. We've got features that are these peaks, algorithmically identified peaks, and we count how many reads were identified in each of those peaks in each cell. It gives us another gigantic and extremely sparse matrix, where this time the features are peaks rather than genes. And so this is sort of the input data for a single cell multiomics analysis. It's huge. We're often dealing with hundreds of, over 100,000, like 1 to 200,000 peaks, 25,000 genes, and tens of thousands of cells. So there's a huge amount of data here. And I'm not going to get into every aspect of the analysis, but it's super, super complex. It's a multi-step process, depending on when you choose to combine the data sets. There are different ways of going about joint embedding and doing clustering and doing differential expression and pathway analysis and trajectory analysis and all of this fun stuff. My student, Chun Li, is sort of our expert on this type of analysis. But I want to talk about specifically one aspect that we're focusing on, which features the Kidney Precision Medicine Project. We've got a really nice little working group of young faculty that's been looking at this data. And specifically, there's multi-ohm data from kidneys from patients with healthy controls and some with kidney diseases. And the interesting thing about this is basically trying to find mappings between the two feature spaces. So I talked about peaks for ATAC-seq and genes for RNA-seq, and they regulate each other, or at least there's a regulatory element to their relationship, because those peaks might be covering an enhancer or a promoter or some other functionally relevant part of the DNA, that then plays a role in how a gene gets expressed. And what a lot of people do when they have multi-omic data like this is just map each peak to its nearest gene. And that can do all right. You can get, a fair number of the relationships that you should identify from that. But what we're working on is using annotation, because there's a lot of genomic annotation out there for regulatory elements, things like enhancers and promoters, that link them to the genes that they regulate. Because the genome exists in 3 dimensions, it folds over on itself. A functional element in the DNA does not always regulate the closest gene in linear DNA space. Sometimes it, will regulate the expression of a gene somewhere farther away. And some of those have been identified, some of those are available in publicly available annotation databases. And so we're using that information to basically infer better gene regulatory networks. How is each gene being affected by these different peak regions? Okay, I'm going to switch gears entirely and talk about spatial transcriptomics, which is another really cool single cell-ish technology, not always exactly single cell. And I'm going to focus on imaging-based spatial transcriptomics. Basically, we can take a thin slice of a biological tissue, put it on a very special plate, and measure the levels of different transcripts at super, super high resolution. And so you can see in this zoomed in image here, each individual cell is outlined, and then we've got little dots representing transcripts of a particular gene. And so you can't do this with every single gene in the genome, but you can do this with thousands of genes. You can do this with quite a few. And thousands of genes is definitely enough to get a good sense for what's going on in any particular system, where you sort of know these are the genes that are going to be playing a role. These are the important genes. So again, for kidney, there are a lot of genes that just aren't relevant because they're only expressed in brain or stomach or somewhere else. And so the multi-omic aspect of this is we're trying to use single-cell RNA-seq and 2 spatial data sets, both coming from the same kidney patients, and essentially combine them in a way that will allow us to impute the expression of all the missing genes. Because for our spatial data, it's a little bit of a delay. For our spatial data, we've got 250 genes. For the RNA-seq, we've got 32,000. We've got all the genes. This is a very, very hard problem. We can do some sort of combination. We can put all of these cells on a similar frame of reference, but actually imputing the expression of each individual gene in each individual cell is a very, very difficult problem. And so far, I have not been able to do this. This represents my best attempt. And you can see the correlation here is not very good. We're not doing a very good job of computing these genes. The nice thing is you can get a general sense of the overall pattern. So this is 3 of those same 6 genes from the previous slide. We've got red is the actual expression, blue is the imputed expression. It's still not great, but you can get a pretty good sense for the pattern of expression in the overall kidney in this case. And then just for completeness, here's the other 3. That delay is getting me, but we can do a pretty good job of getting the overall pattern. And then I don't know how much time I have. I'm out of time. I was going to talk about pattern recognition, new project that Lou and I are very interested in, just recognizing patterns in this data and trying to identify spaces where that pattern occurs. Okay, thank you very much.
While we're switching here, any questions? Because I do have a question.
I have one too.
Oh, great. Howard, go ahead.
Oh, so would adding DNA methylation data give you some sense of the relationship between the RNA and the peaks that are there? In other words, the genes are actually being turned on or turned off or regulated in some way?
Yeah, I think it would. I think all of these are important layers. Right now we're focused on RNA and ATAC because that's the technology that was most recently put out by 10x Genomics. And so it's like commercially available. People can do both of these things together very easily. Methylation and expression, or methylation and DNA accessibility, are a little bit harder to combine. And so that's not a thing that as many people are doing.
Got it.
Okay.
Great, thank you for the question. Last, just to Nick, as you come up, and so you were showing sort of a single individual. What complications across individuals?
Oh, boy. Massive.
Because that's where everything's
Yeah.
going, right?
Yeah, exactly. So the biggest problem is always batch effects with any sequencing. If you run same humans, same sequencer, even on the same day, like you'll still get massive batch effects between runs. So yeah, you have to think about it very carefully. I don't have a quick answer.
Okay, no, that's a great short answer. Okay, next up, Nick Mancuso.
Cool, thanks, Dave. Folks can still hear me online? Okay. All right, awesome. So my name's Nick Mancuso. I'm faculty here in Biostats. And so what I plan to talk about is really just a high-level overview of sort of the directions that or like the primary lines of inquiry that my lab is working in. But really, so my group is like a computational genetics group, maybe statistical genetics, I don't know, population genetics, machine learning, I don't know, whatever. You just throw it all in there, and it's like on Kelly's slide, we went from the like milkshake to the or whatever the smoothie to the fruit. It's just we're going the other way. Just blend all those topics in. But really, we can at least like delineate between two broad things that we focus on. One being mechanisms for complex disease through the lens of genetics, and the other being this notion of genetic architecture and natural selection. And so, if those aren't clear, hopefully, we'll sort of at least shed a little bit more light on them going forward. Okay. So just to provide a bit of background, so global biobanks are ubiquitous now. I think researchers now are really in a fantastic place in which we benefit from the availability of large-scale genetic and phenotypic data sets that are accessible just through an application, right? Like we don't have to write our own grant to collect these individuals. Certainly, there's still a need for that, obviously, for more specific, like maybe biospecimen collection. But if what we care about is maybe like outcome with respect to genetics, there are already millions of participants across multiple biobanks globally. And so here, this is like already, this is a reference is outdated, but back in 2021, we're already at more than 2 million individuals in which there is both genetic data and like rich phenotypic data. When I say that, it's not just like a single phenotype that's measured. You can think about like electronic health records, like really just a vast catalog of things that have been either measured or determined through some sort of like electronic charting technology. Okay, and so with that in mind, one of the big things that my group is trying to dissect is this notion of an evolutionary architecture for complex traits. And so, what the heck does that mean? Well, broadly, when we think about this notion of a genetic architecture, really that's just like a fancy term for characterizing the distribution of genetic effects, right? You can think about like characterizing some parameters or some parameters for a family of distributions that describe how the effects of genetic variants relate to some disease or some trait of interest. That's been generalized over the past 10, 12 years to this notion of a functional architecture. And so now, rather than just having this sort of singular effect size distribution in which effects, are spawned out of in some like generative sense, we know that different properties of variants dictate what their effect sizes would look like, right? And so what that means is if a genetic variant is, overlapping multiple ATAC-seq measurements, we are a priori would think it would have a larger effect size in that particular cellular context than something that didn't, right? And so we can sort of further generalize that notion of like, okay, not only do we have just a single distribution, but we have a higher sort of like a family of distributions that are parameterized in some way, shape, or form through some functional annotations. Additionally now, what we think of as well, that's all well and good to get a sort of a mechanistic and cellular sense, but we can further overlay additional information that relates to evolutionary processes, right? Natural selection does its best to try to prune deleterious variants from the population. And so if we can incorporate any sort of evolutionary information, this further gives us information about how we can sort of describe broadly the effect sizes for a trait or disease. Okay, and so one of the main focuses of my group is to infer these evolutionary architectures. Maybe I should have cleaned some of these words from nowadays, but characterize evolutionary architecture in diverse populations. So a lot of what we do is focused on genetic data across different continental populations to do a better job about identifying what variants underlie disease risk. And additionally, a lot of our efforts are focusing on characterizing this evolutionary architecture in molecular data. So rather than the outcome of interest being, let's say, like prostate cancer or height or BMI, it's a bit more closer to the metal, where what we're looking at are things like protein levels or gene expression. Because ultimately, if we think disease risk or quantitative traits are some end product of those regulatory processes, if those end products have selection acting on them, that must percolate downwards to selection acting on those, again, like molecular phenotypes. And so a lot of the efforts in my group is focusing on that. And additionally, we can think about like incorporating newer sequencing technologies like Return-seq, which is sort of like a broad scale, like a genome-wide scale perturbation assay, where you can imagine like knocking out variants using CRISPR, like genome-wide, right? Like maybe like 7,000 variants or 20,000 variants get knocked out, and you can measure the consequences of that knockout in RNA. And that gives, again, a sense of what would the effect sizes look like. Additionally, so it's not, a lot of the efforts in my group isn't just like highfalutin evolutionary stuff. Bringing it back to some very practical matters, we work a lot in prostate cancer, or the genetics of prostate cancer through collaborations here and elsewhere. And so one of the things that we're doing is we're generating a large amount of molecular data in diverse ancestry individuals, where we'll have mRNA, protein levels, metabolite levels together with germline genetics. And so by characterizing how the germline genetics correlates with all those molecular modalities, we then integrate those findings with downstream prostate cancer risk to, again, prioritize like what genes may be underlying risk, and what other sort of kind of across the entire molecular cascade, and how that leads to disease risk. So that's kind of like the big picture science, but a huge aspect of what my group does is also Bayesian analysis. I am somewhat of a pragmatist about this, and that I'm not saying like you have to be Bayesian to be in my group, or like you have to be Bayesian, or Bayesian's the right way to analyze. Like, it's use the tool, whatever tool works for whatever your setting is. That's. But while we're here, I'll do a little bit of proselytizing for this. And so for Bayesian analysis, it provides a nice framework for quantifying the uncertainty in our estimates, conditional on data. So that's just kind of like the definition, but the great thing about it is it provides like a rigorous framework for how we can integrate prior information, right? Whether that's from functional information, evolutionary information, all that stuff can go into our inferential tools. And so I've presented this slide in numerous talks. This is like, if you Google Thomas Bayes, this is like what comes up. He didn't look like this. This is just some drawing that someone did at some point. Like, we have no idea what he looked like, but that's like just some old sketch.
That's the prior.
Yeah, that's right. That's right. Yeah, exactly. Right.
I mean, data.
Yeah. That's right. And not only that, but like the formulation that we're used to is not even what Bayes came up with. It's due to Laplace. So I don't know, it should be Laplacian inference, whatever. That's where we're at. But the problem is that, most folks know that oftentimes Bayesian inference is computationally hard, right? Like we have these practical limitations in that we can't really solve this, can't really compute this denominator easily. And so what can we do? There's this very useful but very annoying approach called variational inference. I say annoying because it's just a lot of tedious calculations. But if one can sort of force themselves to work out the calculations necessary, it provides a means to turn this sort of integration, right? Like when we think about Bayesian analysis, oftentimes we're doing integrations or integrals or something like that into an optimization task. And so this optimization can be done much more quickly. We're not getting anything for free. Essentially, what we're doing is we're approximating the Bayesian analysis that we want to do by minimizing some sort of sense of distributional distance. I'm pretty over, so I won't go into details. But if this sounds interesting, I'm always happy to chat. We'll skip the math. But we collaborate with a lot of folks here at USC and elsewhere for folks who are working again, mostly in genetics applications, genetics or genomics applications. That's it.
Thank you. Any questions for Nick?
All right. Thanks, all.
Okay. Thank you. Next up will be Josh Millstein.
Right.
That's you. Let me make sure we don't have anybody in. Ready for. Okay.
Okay.
All you, Doctor.
So, my research in terms of methods development is mostly focused on multi-omics, so problems like mediation analysis, false discovery rates, which is large-scale hypothesis testing, dimensionality reduction. But today I just want to focus on a specific analysis that I've been working on, which I think is really exciting. And it's a collaboration with a colorectal cancer lab that I often work with. It's a lab of Heinz Joseph Lenz in the Norris Cancer Center. And lately, we've been interested in the microbiome and its role in colorectal cancer progression and response to treatment. So we have access to a lot of different clinical trials. One of the largest, which is a phase 3 colorectal cancer clinical trial, focused on two specific therapies, bevacizumab and cetuximab, and then also the combination of bevacizumab and cetuximab. So this was a large trial involving over 1,000, quite a bit over 1,000 patients. And in addition to answering that primary question of which therapy is better for these patients, which it actually turned out inconclusive, but a lot of molecular data were collected. So molecular data from tumor samples from these individual patients. Now we're interested in the microbiome in the colon, but also in the tumor itself. So bacteria are throughout, they exist throughout our body, on our skin, in our colon. They also exist within tumors, within cancer tumors. So in the 80405 study, they didn't actually collect microbiome data. However, it's been, various groups have found lately that in RNA-seq data that have been collected specifically from human tissue to learn about human gene expression, as Kelly was discussing earlier, there is what's thought of as contamination. So there are a lot of methods to focus this data specifically on human genes, but they're not perfect. So there are sequences of bacteria that get in there. And what we developed a pipeline based on what some others have done recently to isolate those isolate those sequences and be able to calculate abundances for different bacteria and other members of the microbiome. So this is an overview of the pipeline. So tumors were collected in over 500 patients, preserved in formalin-fixed, paraffin-embedded tissue. RNA was extracted and sequenced, and then it was aligned to the human genome. So there's a recent genome that is much more comprehensive than earlier version of the genome, and it includes a lot of repetitive regions that are important in the characterization of the microbiome, because if we don't, so the process here is we map the reads to human first, then we isolate what was considered contamination and then map those to bacteria. But we have some regions of the genome that is where human and bacteria are the same. So we have to first get rid of all the sequences that map to bacteria. So then once we have those isolated contaminants, essentially, we can map them to bacteria and other types of microbiota, and then calculate abundances in patient tumors and relate those to clinical outcomes such as survival. So I went through that process, developed the data, identified over 2,700 taxa with some evidence in the tumors. But what we're really interested in is those taxa that are represented in some meaningful proportion of patients. So we focused on taxa that occurred in at least 10% of patients, which ended up being 117 taxa. Then I conducted Cox proportional hazards regression to relate those abundances to survival, overall survival and progression-free survival in the patients, adjusting for patient characteristics and treatments. And what I found overall in those marginal association tests was essentially nothing that met the FDR corrected significance level. So initially that was a little bit of a disappointment, but then I looked at, well, maybe there's an interaction between bacteria levels for some of these taxa and treatments. So it could be that taxon is important in one treatment but not another, or has a positive effect in treatment A and a negative effect in treatment B. So I fit an interaction term here to look at that, and this generated several interesting taxa. So two of which met an FDR correction level of 0.05 that I'm just calling CK and MT, and the other that was close at a FDR level of 0.058. So if we look at Kaplan-Meier survival curves for patients that had the bacteria in their tumors and those that didn't, what we can see here for the CK bacteria is that patients with the bacteria did worse. So overall, among both treatments, this is the top row. On the left is progression-free survival. On the right is overall survival. So you can see that the patients with the bacteria in blue did worse. But if we look down at the treatment-specific curves, so on the second row it's bevacizumab, it's a much bigger effect. So those patients did quite a bit worse. In cetuximab, there was no apparent effect at all for progression-free survival. So there's really quite a discernible difference here between the treatments, bevacizumab and cetuximab. And that's interesting because there are several practical things that could come out of this. For instance, we could just by measuring the microbiota in the tumor, which was sampled prior to treatment in this study, we could know that these particular patients with the bacteria won't respond well to bevacizumab. So it's actionable information immediately. Also, there's a potential intervention. There are antibiotics that actually have been developed and are known to target this particular species because it happened to be found important for other indications. So, and then with the other two species, it was actually the opposite effect. So still focused on bevacizumab with not much effect in cetuximab, but here the bacteria is beneficial. And the implication is, again, if they have this bacteria, they would respond well to bevacizumab, probably. Also, it raises a possibility of whether we could develop a probiotic and actually a treatment for these patients. And then the last one, AE is a similar pattern that it seems to be beneficial to the patients. So if we look at another outcome, which is referred to response or RECIST response, that measures whether the tumor expands or shrinks in patients. So CR is a complete response where the tumor shrinks to nothing, essentially. PR is a partial response where it shrinks but not to nothing. SD is stable disease, and PD is progressed disease, so it increases. And what you see here is on the top level with CK, there's not a lot of difference between the left, which is bevacizumab or cetuximab, and the right, which is bevacizumab. But if we look down at MT, in the bevacizumab, but not the cetuximab, there's a really compelling difference between complete response between those who have the bacteria and those who don't. So that it doesn't necessarily follow because the response doesn't always correspond with progression-free survival. So this is additional evidence of the efficacy of these microbiota. And I don't know how much time I still have. 1 minute. Like 1 minute? Okay, I'll just kind of wrap up that we looked at gene expression as well. So relating the microbiota to gene expression. And what we found here is a significant difference in the distribution of p-values between the associations with microRNA and coding genes, suggesting that this particular taxa, the MT, this taxon, may affect microRNA levels, which are regulatory in a cell. And I conducted mediation analysis with an approach, a hypothesis testing approach I developed called the CIT, and identified a gene, the CDC37, which we actually published on last year, that is consistent with mediation for the CK taxa. So it depresses expression. So the bacteria depresses expression of this gene, and the gene low expression is associated with poor survival in patients treated with bevacizumab, but not cetuximab. And that's where I want to leave it. Josh, any questions that we transfer?
Chun, do you want to? One question from Howard.
Sorry.
No,
So
no.
your last slide kind of addressed my question, which was, what's the biological mechanistic basis for how the microbiome might actually modify, tumor response to chemotherapy. But has that been sort of been supported by direct evidence of, somehow the bacteria causing the gene suppression or enhanced expression?
Not a lot is known at this point for that particular taxa. It's known to cause a pathological infection in certain cases, but in terms of the mechanisms that could be going on within the tumor, that's a huge question, and we really don't know. We're trying to just chip away at it with looking at gene expression and stuff, but yeah, we're actually collaborating with a lab that cultures bacteria, and we're hoping to make more progress identifying mechanisms that way with cell lines and stuff and
Very
culture.
cool. Very cool. Thank you.
Thank you. Okay, next up, in our speed talks, Chun Li.
Thank you.
There you go.
So I know the times for results. I try to do it in within maybe 5 minutes.
Okay.
The title is misleading, and that just is a generic one. Yeah. So I came here 5 years ago. I'm teaching 2 courses now. I'm interested in general biostat, stat gen, and machine learning deep learning. So I'm going to spend time on this specific topic that's really interesting for at least we started about 10 years ago. We essentially want to analyze continuous data, continuous outcome, often skewed, especially in biomedical research. And sometimes mixed. Regression often requires extra attention, which is if you just naively put Y there and regress on X, you have problems. So oftentimes transformation is needed. But the selection is often ad hoc. For example, often people choose logarithm or square root, which is okay. Certainly, oftentimes better than not transforming the outcome. But we don't know whether it's correct or not. But it's certainly better than not doing. A fancier way is good to do so-called box-Cox transformation, which is just a family of power transformations. So it's really not covering all possible transformations. And there's almost nothing beyond that. So the problem is, if you don't know what the transformation is correct, if you just pick one, you just stay with them. And whatever your conclusion comes from it is depends on the choice of your transformation. And sometimes different transformations may just give you a different results. So this problem exists everywhere, actually, as long as you deal with continuous outcome, whether you do a traditional statistical analysis or you do deep learning, you have the same issue. So,
All right, everybody. This is the complication. I'm screen sharing now. There we go. Now they can see your screen.
oh, okay. Sorry, folks, that you didn't see it. All right. So I already went half of the talk, So, all right. So our solution is to do instead of picking the transformation, we want to estimate it, and we estimate it non-parametric. So we came up with this thing. We call it CPM. But basically, it tried to do rank-based parametric regression analysis. It's written this way: if you have learned ordinal data analysis, you actually recognize the similarity between this and ordinal data analysis. It turns out that what we are doing essentially is to treat outcomes, continuous outcomes, as if they're ordinal. But we have justifications for why we do it. There's another way to motivate this analysis is to start with something called a linear transformation model, which is, instead of directly connecting Y with X in a linear way, which is strong, you can think about the latent variable called Y star you never observed, and Y star connect with X in a nice linear way, but the connection between Y and Y star is monotonic, but with transformation unknown. So, and this has been studied for decades in survival analysis. It turns out these two models are equivalent. And the intercept function here we are estimating is the transformation that's needed. So for example, logarithm or whatever other transformations you may choose, instead of choosing them, you could just estimate this transformation non-parametrically. We did some, work on this. So get a few papers out. One paper was last year in JASA, basically saying this method could be used for detection limits. And last year, I gave this talk for our seminar and got a few faculty here interested. Kelly reached out to me saying that, basically skew data is really notorious, widespread in the transcriptome data. And people always talk about transformations, and again, different transformations may lead to different conclusions or different top list of candidate to follow up. So, and seems to be a method that may work. So one of his students did some simulations, and the latest results seem to be this method, which is really not designed for transcriptome single cell analysis, is really a method pulled from the shelf. Essentially perform almost the same as the state-of-the-art method designed for transcriptome analysis. So that's good message. And then also, David also mentioned that there are other omics data that really could potentially benefit from the same thing, because we always talk about transformations and which transformation is correct. All right, so now it's half. So the next three slides basically talk about potential directions we are working on, and currently working on, and future directions. One is certainly we want to relax this beta X format in the regression. One direction is to relax it so that especially when your sample size is much bigger than your number of parameters, you have enough information to learn more subtleties, more details. And so currently we're working on a random forest method on it. But in the future, we are really planning to do boosting. And I put a question mark on deep learning. It's essentially the same question there. How would we do it? I haven't thought about it. Another direction is in the situation that you have your sample size is not very big, like traditional statistics, but you often have many covariates, like dozens or even hundreds of covariates. So in this case, you cannot throw all the variables into your model because your model is going to be unstable. And so oftentimes we want to regularize like ELSNet or last order enriched regression. So currently, Victor and Eric are working with me to implement method algorithms for this one. Another direction of research is, as I mentioned, that in survival analysis literature, people studied this kind of thing for decades, and they all know that Cox proportional Hess model is a special case of it. And our JASA paper last year showed that for detection limits, if you have detection limits on both sides, this method actually works better than existing survival analysis method that designed for W sensor data. And Frank Herrick, which is was a chair at Vanderbilt, and he developed a lot of R packages. And he recently discovered that CPMs can be used to perform quite a few other popular survival analysis. So essentially, CPM, essentially this whole thing could potentially be a new paradigm for survival analysis, and we're investigating it. And the side note is, we have something called probability scale residual, PSR, and Eric Kawaguchi is working on the manuscript revision to extend it to various censored mechanisms. So this would be also useful in this extension. And the last one is in causal inference. In causal inference, people talk about ATE a lot, or sometimes ACE. They stands for the same thing. ACE stands for average causal effect. Again, like many statistics, ATE is sensitive to skew data and outliers, and can be difficult to estimate when you have mixed data. So alternatively, we could have robust causal estimates like these three. And currently, we are just basically putting the CPM model into estimating these things in a fashion that's doubly robust, like the AI PW estimators or PMLE estimators. We also work on a DID, which is another aspect of causal inference. Well, I'm going to skip this one. This is my collaborations. All three of them involve genetics, actually. That's it.
Any questions? Okay. Jim, do you want to share with, okay, so I'm gonna stop the screen.
I'll share on Zoom.
We'll get this worked out, everybody. Hard with 5 talks. And I'm fully in IT, that's not a good feeling. Okay, all right, Jim Gauderman,
So I'm
Is that-
using mine to go down.
you use yours to that, but volumes and everything's on it. So
Okay.
you're all set.
Oh, it actually got to me this week. I'm very surprised. Thank you to the pre-speakers for being quicker than last week's. Anyway, so I'm going to talk about gene-environment interaction. It's work that involves a lot of faculty, staff, and several of the students who are listed in blue down here. It keys off of something called a program project grant that we have kind of uniquely in our biostatistics division compared to other biostatistics programs. This one focused specifically on statistical methods, and I think it's like the only currently funded methods PO1 in the country. It's focused around integrative genomics. It involves 3 main projects. We call them P1, P2, and P3. I put all the various faculty on here that are leading various projects, and then it also is supported by the computational core, an outreach and education core, a data core, and the annotation core. So if you're interested in any of the topics listed up here, you can note who those faculty are. My project focuses on gene-environment interaction, and if you're not familiar with what that is, you probably are, but you know, why do we want to consider that? It's the situation where we think, you know, both genetics and environment are important, and particularly that there are modifying effects of disease. So why do we want to do it? Well, maybe they're both involved in an oxidative stress pathway. We want to understand biology. We want to just build a better prognostic or prediction model. Maybe there's a genetic component to a disorder like BRCA1 in breast cancer, but we want to see whether it's stronger in one particular subgroup, or maybe an exposure is known to be important, you know, air pollution related to something like asthma. We want to see whether it's stronger in a particular genetic subgroup. So you can think of it as a environmental kind of modified by genes, or genetic effects modified by exposure. These days, we're thinking a lot more along these levels, where we have some environment over here, and as Josh pointed out, that could include treatment in a trial. You have some form of genomics data, could be germline genetics, expression could be microbiome, what have you, and you want to see whether there are modifying effects one way or the other of some genomic factor with some environmental factor in causing some kind of a complex outcome. So a key resource that we have our hands on, we have for a number of years, is called the Functionally Informed Gene Environment Interaction Study. It's focused on colorectal cancer. Very nice collaboration with folks at University of Washington. About 130,000 total subjects, roughly half case, half control. Full genetic data, lots of epi data for a lot of exposures that are known to be related to colon cancer. Limited, but you know, state-of-the-art kind of omics data, and the primary goals of this project are to look for G by E and kind of omic by E interactions. So we, in this group, are primarily focused on methods development, software development, and then downstream applications. So we've developed what's called a 2-step approach. I'm not going to go into any details, except to say that the basic idea is if you have genome-wide, say, SNP data, you can screen that data by doing some kind of a test, in this case, just a test between the outcome and each SNP. You can set some threshold to say, well, I'm only interested in SNPs that achieve a p-value that is at least smaller than some alpha. This is the reverse order, so these are the more significant ones. And I'm only going to test G by E interaction for these SNPs that are above this red line. Okay. The payoff is that then for that smaller set of SNPs, not all the SNPs, you can now get the proper type 1 error rate by just comparing to some other alpha level that is smaller than that, only based on the SNPs that pass that threshold. Again, I don't have time to go into details, We have a lot of papers, a lot of simulations showing that it does indeed improve power. So we have applied this for the last probably 6, 7 years to a variety of exposures in genome-wide scans for colorectal cancer. These are each different papers. They've each been published in top journals. And I've noted the kind of in blue here, the approach, and the ones in blue are ones that our group has developed that found significant associations, which I look at this and I say, well, if you kind of look at this and say, if you didn't find a significant thing, the paper probably would have not got published or not got published in a good journal. So several of these papers were really only kind of saw the light of day because of a lot of the good methods work that happened here. And software, I have to point out that John Morrison has developed, which has really become the state of the art. Several students working in the past and currently, and Bo and Key both working on currently on methods to improve power even more for these two-step and also to do kind of joint modeling of SNPs and environmental factors. 3 minutes. So we've also spent time thinking about polygenic risk scores. You've heard a little bit about this from Nick. Basically, it's taking a lot of SNPs, making a linear combination of them, creating one measure, which gives you an overall general feeling of genetic risk. And a lot of people are looking at things like this, right? Rather than doing SNP-specific interactions, you throw a PRS in there. The problem I see is that a PRS is kind of a mix of a lot of different SNP effects, some of which may have an environmental kind of modifying effect. Most of them probably don't. So this one PRS is probably kind of diluting some underlying gene environment effects. So we've been thinking about breaking that PRS into subsets. And how do you do that? Working with Yu Mi and his group in our annotation core, he has a very nice package for annotating SNPs and identifying their functional pathways. So we break them into pathway-specific polygenic risk scores. We have a paper that just got published reporting this. And long story short, when we look at overall PRS by E interaction, and here E is aspirin and ibuprofen, which is jointly called NSAIDs. We don't see any significant interaction when we put an overall PRS, but when we break it down into pathway-specific PRSs, we see some really strong interactions with particular pathways. So it not only identifies an interesting kind of general risk group based on this is the TGF-beta pathway, it also is an indicator of maybe which SNPs and pathways could be important for how aspirin and NSAIDs affect colorectal cancer. And then I'll conclude with one final slide. So we've been thinking a lot lately, Ming-Ji and Jessica and Juan, and a bunch of us, about deep learning. And then very kind of the cool thing about deep learning inherently is that it finds nonlinear patterns, of which one of those is interactions among factors, right? That's just kind of what it does. So it seems kind of a priori, maybe a no-brainer to think about identifying interactions through some kind of a deep learning model. It's also good at finding nonlinear patterns. So much of the work that we do has been based on standard linear models, Cox models, logistic models, where everything is kind of linear terms in there. Maybe you throw a quadratic, maybe you think to put an interaction in there. But if you have a lot of SNPs or a lot of expression data and a lot of exposures, it's going to take a long time to think about putting all possible interactions in the model and thinking about nonlinearities and all that. So deep learning and other machine learning provides a nice context to think about doing that in general. So Jessica has been doing a lot of simulations, looking at a range of different kinds of models. I've highlighted some deep learning approaches here. This is R squared for prediction, comparing linear regression to other kinds of elastic net, lasso, random forest, boosting, and in general across some different kinds of fairly complex models. One of the deep learning approaches seems to kind of emerge and do the best at predicting. Not hugely surprising, but a little bit reassuring, and it does seem to do better, at least than random forest, and in most cases, kind of right in there with boosting. So we've taken this to the UK Biobank data Here we have a desire to predict what are called biomarkers. So things like your glucose level, your HbA1c level, things that aren't measured in our Fiji data set, but they have been measured in the UK Biobank data. So we can try to build a big model for how to predict a person's glucose level, their blood glucose or their HbA1c or something like this. We have a lot of available data. We have a lot of polygenic risk scores. We have a couple of key environmental variables, sex, age, BMI, et cetera. So we've been thinking about, hey, toss this all into a big deep learning model. And if you kind of look here at the R squares that we're getting, so we have 4 biomarkers we've looked at: IGF, C-reactive protein, HbA1c, glucose. You can see that the prediction R squared on a held out test data set, not used in training, is anywhere from a little bit better using deep learning compared to just standard linear regression, which is kind of what people tend to use, to dramatically better for some of these biomarkers. So, and no difference in pre-processing the data, no difference in what you feed in. You're still feeding in all the kind of main effect terms, just letting kind of the deep learning model find all these interesting possible relationships. And especially when we want to do prediction like this, this is kind of a nice way to go. The other nice thing about a deep learning context, and this kind of brings it back to the idea of integrating additional information, is that we may have SNP data, we may know something about the pathways that those SNPs are in, and it's relatively straightforward, I'll say, to think about building our deep learning architectures in such a way that we can not connect everything together, but connect only SNPs, certain nodes that relate to commonalities and pathways, and then maybe we introduce exposures at that point, and then we're modeling forward. So this is a very simplistic look at how you can think about networks in pieces and then think about patching them together in one huge network where it's all fit kind of simultaneously, and yet still try to capture some underlying biology, or you could be thinking about bringing in expression data or other kinds of data and patching that data into that network structure. So I'm going to stop there.
Any questions for Jim? And thank you all, the speakers, and everyone for coming. See you next week.